ladies. Okay, so I just want to do a little revision on osmosis with you. Okay, I know we only did maybe 2 lessons on it, but let's have a little look at this. Okay, so the main things you need to know about osmosis are the following: you need to know the definition, how it affects cells in the body, the main experiment that we did, how it affects plants, and also food preservation. Okay, now first of all, the definition. So remember, we said osmosis is the movement of water molecules across a semi-permeable membrane from an area of high concentration to low concentration. This could also be considered the diffusion of water, because diffusion is movement of any material from an area of high concentration to low concentration. Okay, and just the important bit, girls, is across the semi-permeable membrane. Okay, now cells in the body. So how osmosis is involved in cells in the body. So first of all, your cells in your cytoplasm, there has to be the same concentration as your blood. If it's not, then the cells become unhealthy and you can get what's called crenation or lysis. Crenation is where the cell begins to shrink when it's placed in really, really high concentrations of salt and sugar, or if you become dehydrated and your blood concentration becomes quite strong. If there's high concentration of salt and sugar in your blood, that means the cell will shrink and it will become useless. It doesn't do anything, it can die off. Lysis is the opposite. Lysis is where you place an animal cell in really low concentrations of salt and sugar. So therefore you might place it in pure water and it takes in lots and lots and lots of water and it will eventually burst or explode. Okay, now this does not happen in plant cells, because plant cells have what's called a cell wall around them, which gives them support. So actually, when you give lots and lots of water to a plant, that's actually a good thing, because this creates what's called turgor pressure, which is the pressure between the cytoplasm in the cell and the cell wall, and that provides support from the cell and prevents this occurring, which is where a plant will begin to wilt. So actually, that is quite important for how plants, how osmosis is involved in plants. So turgor pressure occurs in plant cells, gives support to the plant because plants don't have skeletons, and it helps the plant remain upright, meaning they can get more sunlight and photosynthesis can occur. Okay, now talking about the experiment itself. So remember, we took 2 viscine tubings, we placed one with pure water, one with salt or sugar solution, and we left it in a beaker of pure water overnight. Then we weighed them, and what should have happened was osmosis occurred in the salty or sugary solution, meaning that this one increased in mass, which means you weighed it before and after, and this one is heavier. Okay, so that's your osmosis experiment. A very important word, girls, is isotonic. So you might see this on the side of Lucozade. So isotonic is where a solution has exactly the same concentration as the blood, and this is kind of a term that we use kind of in chemistry a lot when we're talking about concentrations of the same solutions of the same concentration. Okay, now so we've talked about turgor pressure and why it's so important for plants and why osmosis is important for plants. Osmosis also occurs in the roots of plants, and root hair cells, we'll do this in plant when we do the plant topic, contain high amounts of sugar and salt solution, which encourages water to enter from the soil, and then it travels up the plant through xylem, that tissue that we talked about junior cert. Okay, so osmosis is very, very important for plants for turgor pressure and also for the entry of water into the roots. Now, in terms of food preservation, there's 3 ways that osmosis is related to food preservation. So there's salting, sugaring, and pickling. So salting is where you can buy like salted meats or salted fish in European shops or Asian shops. The salt means that the bacteria cannot grow on the food because it will crenate. So the lack of water there and the high levels of salts means that the bacteria lose water and therefore they cannot grow, so that fish and that meat will last a long time. Same with sugaring, girls, which is how jam lasts a long time. So it's a way of preserving fruit. So you put fruit in a really high sugar concentration, makes a sugar jelly, and that very little bacteria, viruses or fungi can grow on that, because the high sugar concentration means that bacteria, viruses and fungi will crenate as well. Then pickling, girls, is actually slightly different, because it means that the low pH or the acidic nature of vinegar means that nothing can grow on the food, therefore the food lasts a longer time. Now I just want to have a little look at some exam questions on this. So we're going to have a look at down with the plants, because it generally comes up with plants. Now, osmosis and transport. Okay. Now, so osmosis and transport questions. Now, so Let's make that into a bigger screen. Okay, now, so let's have a look for osmosis. Okay, that's a plant-based one. That's a plant-based one. Okay. Okay. When carrying out an activity to investigate osmosis, why did you use visking tubing? Visking tubing, girls, was our semi-permeable membrane, so that allows the movement of water, but it doesn't allow the movement of sugar or salt. How did you know osmosis had taken place? Because the visking tubing with the sugar or salt had increased in mass. Next question. Okay. Again, a lot of when demonstrating osmosis, what did you use as a selective permeable membrane? You used the visking tubing. What did you conclude that osmosis, or what led you to conclude that osmosis occurred? The one with the salt or sugar had increased in mass. What do you understand about turgor pressure, or what do you understand about the term turgor in plant cells? This is the pressure that occurs due to the cytoplasm having enough water in it to support the cell wall. Therefore, this supports the plant. How do plant cells remain turgid for a considerable period of time? They take up water through their roots, and then the water is provided to the cytoplasm of the plant cell. What happens to a plant if its cells lose turgidity? It will wilt, which means it falls over. If an animal cell was subjected to the conditions that result in a plant cell becoming turgid, suggest what may happen to an animal cell. It would explode, girls, okay? Because turgidity requires a lot of water. And if animal cells take in a lot of water, they don't have a cell wall, which would support it, so therefore they undergo lysis. In relation to the passage of materials through cell membranes, distinguish between diffusion and osmosis. Diffusion is the movement of any material from areas of high concentration to low. Osmosis is the diffusion of water across a semi-permeable membrane. In the study of your human physiology, you encountered an example of active transport. Name the location and the material involved. That's related to the kidney, girls, okay? Okay, so let's skip on to the next question. Let me see, there we go. Okay, so in relation to structures such as cell membrane, explain selectively permeable. This is a membrane that only allows certain things to cross, such as water. It doesn't allow larger molecules. Suggests an advantage to the cell of having selectively permeable membrane. It prevents larger, more dangerous substances from entering the cell which could damage it. Name 2 substances that enter human muscle by cell diffusion. Well, human muscle has to undergo respiration, so therefore you need oxygen and you need what's the other thing? Oxygen. Well, water will also enter by diffusion, and probably glucose as well. Explain the term turgor. Turgor is the internal pressure that supports a plant cell. Give a feature of a plant cell that allows it to remain turgid for long periods. It has a cell wall which supports it. Suggest a way which turgor is of value of plants because it allows the plant to stay upright and therefore undergo more photosynthesis. When demonstrating osmosis, what purpose did you use the visking tubing? This is where the semi-permeable membrane. At the end of the demonstration, how did you conclude the osmosis had occurred? The visking tubing with the salt or sugar solution increased in mass. I think that is all the osmosis questions there. There's also a number of osmosis questions that occur under the experiment questions too, which we'll do in class the next day. So girls, that is osmosis, and I hope that was